You're welcome back to a continuation of our online lecture on cytology. If you would recollect, we already discussed the cell theory, where we talked about the rules of Robert Hooke, Matthias Schleiden, as well as Theodor Schwann. In the continuation of our lecture today, we shall be describing the animal cell. Of course, as I illustrated, the cell is the basic, structural, and functional unit of life. So, by describing the animal cell, we are describing the structure of the animal cell. So, the animal cell structurally can be classified into three components. We have the cell membrane, which is the outer portion of the cell. The animal cell is a structure that looks like this. So it has the outer component, which we describe as the cell membrane. Then we have the internal component, which is referred to as the cytoplasm. This cytoplasm, like I stated earlier, is different from the cytosol. The cytosol is the solid fraction of the cell. The part of the cell that houses most of the organelles is the cytosol. The cytoplasm is more of the fluid portion of the cell. And in most type of biological reactions, such as glycolysis and the Krebs cycle, glycolysis mainly occurs in the cytosolic fraction of the cell, while the Krebs cycle occurs in the mitochondria. So we have the cytoplasm, then we have a more central portion. This is quite a distinction between the animal cell and the plant cell. Unlike in the plant cell, you realize that the nucleus is peripherally located. So if you look at a plant cell, the nucleus of a plant cell is more located towards the edge. But looking at that of an animal cell, the nucleus of an animal cell is centrally located. So this is the nuclear envelope. So back to my illustration, the animal cell is comprised of three major parts: the cell membrane, the cytoplasm, and of course the nucleus. The cell membrane is going to be a topic on its own, but today I'm going to be giving you the cellular organelles, how it is drawn, and then finally I'll be rounding up by giving you the transport across the cell membrane. So this is the animal cell. As you can see, it is enveloped by what we refer to as the endoplasmic reticulum. So the nucleus forms an extension known as the endoplasmic reticulum. So this is basically how your cell, your plant cell, your animal cell looks like. So this is more like an enlarged structure of your animal cell. Then looking at the internal portion, there is a thickened area inside of the cell. This thick in portion is what we refer to as the nucleus. The nucleus. Please, candidates tend to make a mistake between the nucleus and the nucleolus. This shaded portion is the nucleolus, but this portion inside here is the nucleus. Then the membrane surrounding the nucleus. This is a membrane, this is another membrane, this membrane. This membrane that surrounds the nucleus is referred to as the nuclear envelope. They are called the nuclear envelope. Some literatures may describe them as the plasmalemma, the nucleolemma rather, nucleolemma, or we call it the nuclear envelope. So it has three names. Either call it the nuclear envelope, or you call it the nucleolemma, Is that how you call it, the nucleolemma? Nucleolemma, or you call it the nuclear envelope. So the nuclear membrane is divided into three. The nuclear has three names, the nucleolemma, and its formed extension. This extension around the nucleus are called endoplasmic reticulum. This extension, they are looking more of like a finger-like structure. Around the nuclear envelope, we call them the endoplasmic reticulum, for short, ER. So ERs can be divided into two. We have, of course, we have discussed the nucleus, we have discussed the nucleus, we have discussed the nuclear envelope, we have discussed the endoplasmic reticulum. Each of those structures I will be describing their function. Primarily, we have the ER, then of course, we have, it can either, I said we have endoplasmic reticulum, which can either consist of ribosomes. Look at the dotted structure. Here we call them ribosomes. And the possession of ribosomes, so ER, we can state that ER is divided into two. That is, the endoplasmic reticulum is divided into two: ribosome possessing and we have the ribosome lacking. When the ER possesses ribosome, when the endoplasmic reticulum possesses ribosome, it is referred to as rough endoplasmic reticulum, RER, rough endoplasmic reticulum. However, when it lacks ribosome, we call it smooth endoplasmic reticulum. So endoplasmic reticulum are basically divided into two. They are of two types rather. We have the smooth endoplasmic reticulum, which lacks ribosome, and we have the rough endoplasmic reticulum, which is ribosome possessing. So I've illustrated that, talked about the nucleus, which is the most innermost structure of the cell. This is where you find your chromatin, your DNA, and all other genetic components of the cell. So when we look at the nucleus is usually described as the master cellular organelles, because the information that are required for the synthesis or the manufacture of other cellular organelles are contained in the nucleus. Then it is also the site of cellular division. So the cellular division occurs primarily in the nucleus. So I've discussed the nucleus, I've discussed the endoplasmic reticulum. I've said there are two types of endoplasmic reticulum. We have the ribosome possessing, that is the rough endoplasmic reticulum, and of course we have the ribosome lacking, which we refer to as the smooth endoplasmic reticulum. What are the primary functions of endoplasmic reticulum? Endoplasmic reticulum, as I've illustrated, has two functions, depending on whether it possesses ribosome or it lacks ribosome. The presence of ribosome in endoplasmic reticulum allows the endoplasmic reticulum to be able to manufacture proteins. Ribosomes are consisting of RNAs that enable protein synthesis. Hence, the rough endoplasmic reticulum basically facilitates the manufacture of proteins. So, rough endoplasmic reticulum synthesizes proteins, while smooth endoplasmic reticulum synthesizes lipids. Rough endoplasmic reticulum are for protein synthesis, while smooth endoplasmic reticulum, they are for lipid biosynthesis. So any lipid biosynthesis, or sometimes even playing a role in calcium homeostasis. So nuclear envelope, the ribosome, I've talked about. So this one's endoplasmic reticulum, you can see them, they don't have ribosome, so we call them SER. Then you can see this, they possess granular structures, which we refer to as, so these ones that possess granular structures. We refer to them as rough endoplasmic reticulum. Then another structure that is common here is the vacuole. This is the vacuole. This is the vacuole. So we must know that the vacuole in animal cell is relatively smaller than the vacuoles that are present in plant cells. So this is the vacuole. Then you could also see substances that look more like the endoplasmic reticulum, but they are not entirely like the endoplasmic reticulum. You see them looking like this. They have a flattened surface and they have sacs, sac-like structure on them. When you see structures with sac-like structure like this structure are referred to as the Golgi. We call them the Golgi apparatus. The Golgi apparatus. Or we could call them the Golgi body. They are made up of a flattened sac of structures known as what? Known as cisternae. So this is the Golgi apparatus. This sac-like structure, which actually are responsible for embedding proteins, for embedding structure, we call them Golgi sacs. So looking at this, we have the Golgi sac. The Golgi sac. So all those sac-like structures are the Golgi. So the Golgi body and the Golgi apparatus is like the warehouse of the cell. So the Golgi bodies or the Golgi apparatus are described like the warehouse of the cell. And what do? Their function is primarily to package, store, and distribute materials within the cell. So any protein that has been manufactured by the smooth endoplasmic reticulum is packaged, transported, and distributed by the Golgi apparatus. So as you see that in most cellular structure, you will see that the Golgi vesicles are very close to the endoplasmic reticulum. Another primary structure that you will see, which is very important in the animal cell, is the mitochondria. It looks like a sausage. The sausage-like structure is a double membraned structure. It's double membraned, and it has what we refer to the internal portion here. This structure, which looks like a zigzag, is called a cristae. But this is the mitochondria. The mitochondrion is more of the energy house of the cell. It's more of the energy house of the cell. So these two structures is the mitochondrion. So we could see the nucleus, the nucleus, the vacuole. We could see the mitochondrion. The mitochondria. See the mitochondria, these two structures, and the mitochondria. Mitochondrion is singular, while mitochondria is plural. You have other structures like the centrioles. We have the vacuoles, we have the nucleus, we have the SCR, we have the mitochondrions, we have oil droplets. These are oil droplets, these are not really organelles. And of course, we should also take note of the exterior portion of the cell. This is the cell membrane. This is animal cell. So I've illustrated the basic components of the animal cell. We've talked about the ribosomes, which are responsible for protein synthesis. We've talked about the nucleus, which is described as the master cellular organelle and controls the function of all other activities of the cell. We've talked about the mitochondria, which is the energy storehouse of the cell. It is the important side of cellular respiration. We talked about the smooth and rough endoplasmic reticulum, which are primarily responsible for protein synthesis as well as lipid biosynthesis. Then the Golgi bodies and the apparatus, which are primarily responsible for transport, distribution and export of materials in and out of the cell. I believe with this explanation we have given to you, we have been able to successfully demystify the concept of the animal cell. Please ensure to like, subscribe and pass your comment. Do have an amazing week. Cheers.